Hi, everybody. My name is Jiries Meehan-Atrash. I'm a fourth-year graduate student at Portland State University in the lab of Dr. Robert M. Strongin. I actually used to work for High Times magazine. I was their science editor, and after studying, getting my BS in chemistry. And so I started doing, I joined Portland State University, and they had been doing this work with e-cigarettes for a fairly long time, maybe around 5 years. And before that, they were doing a lot of work with tobacco. And I joined and I said, hey, why don't we extend this project over to the cannabis vaporizers? And so that's when I started working there. And it's really been a great experience working with these people here. There's a lot of really intelligent and really hard workers here at Portland State. And we're doing a lot to uncover some of the chemistry that's going on with these vaporizers. And really, I think that one of the strengths at PSU here is that, we're not just about, quantifying components in here, but we actually look at the mechanism of how these degradation products are forming. So there's a lot of really interesting chemistry here for anyone that's interested. And so my presentation, I'm going to be talking about some of these things that we've found out about the vaporizing of cannabis components that are typically put into vaporizers or used for dabbing. And so, but I'm also, because this is sort of for credits, and this is something that is meant to be a little more educational maybe than the standard presentation, I am going to be going over some background on aerosol science, to put it generally. And yeah, I think this should be pretty informative for anyone that's interested in this kind of emerging field in cannabis. So a little background on cannabis botany. I would imagine that, given the audience, most people aren't too unfamiliar with what's on this slide, but I'll go over it. The essential oils, which are going to contain the active ingredients of cannabis, are located in external structures called trichomes, and you can see a photo there. They're pretty small, as you can see from the animation. You zoom into a cannabis bud. They're these little tiny structures that contain the oils, and these are going to be what contains the THC, the tetrahydrocannabinol, the active ingredient, or cannabidiol, CBD, another active ingredient, and other cannabinoids as well. And also the terpenes are going to be located in these trichomes as well. All other parts of the anatomy of the plant contain around 3% cannabinoids for cannabis plants that actually produce a lot of terpenes. And so really it's all the outside. And so what people do is they take these buds, dry buds or freshly harvested buds, just the flowers mostly, and they put them into containers, stainless steel, what have you. They pass solvents over them. These can be butane, supercritical carbon dioxide. And these are solvents that are nonpolar because the active ingredients, the essential oils, are also nonpolar. So like dissolves like. So you have your butane here. Supercritical carbon dioxide is a nonpolar solvent. It actually has a polarity roughly around that of hexane. It's also used to decaffeinate coffee. It's one of the ways that people decaffeinate coffee. And what people have been doing maybe in the last 5 years or so, it's getting really popular, is people take this kind of crude extract. They use fractional distillation, vacuum distillation, separate out the cannabinoids and the terpenes from kind of everything else, maybe waxes and lipids that are also in there, and then they can recombine these things, or they can take terpenes derived from other sources and combine them into THC and put them in vape pens, for example, right? So I'm going to go over what some of these components are. Now, you can see the set of structures on the right here. Pardon, I can't really point to anything, so just going to have to bear with me a little bit. Cannabinoids start off in what's called their acid forms, and you can see up there CBDA and THCA, delta-9 THC. That delta-9, for anyone that's curious, refers to the placement of the double bond on the upper left-hand most ring up there. And so delta-8 THC is when that bond flops over one space to the left, and then there's other ones as well, much less common. Delta-9, that placement of the double bond there is the most common one, and that's what the plant actually makes, the biosynthetic pathways in the plant actually makes THCA, delta 9, and CBDA. And so these things, these acid cannabinoids, as they're called, are not directly active. Studies, like, for example, I could think of maybe one where these are injected into a mouse into the bloodstream. They don't really do anything. Or if you were to orally ingest THCA, you wouldn't get high because that THCA, not only does it get broken down by the liver pretty extensively, more than THC, but it also, I don't think it crosses the blood-brain barrier. It's not psychoactive, right? And so through a process called decarboxylation, that acid group on the ring is lost. So this decarbing, as it's called colloquially, is something that's done to make edibles. And it's also something that happens, as you might imagine, when you vaporize or smoke cannabis that contains THCA. And that active THC that can now cross the blood-brain barrier is going to be what causes that psychoactive effect. Likewise, this also happens with CBDA. When you heat it, it turns into CBD. And you can see here the way I set up these arrows. The arrows that are pointing down are, you have the H nu, or like that nu is a Greek letter, kind of looks like a V. That refers to light, ultraviolet light. Energy is going to hit those molecules when the plant's growing or on storage, and you're going to have that double bond isomerization. Now that's where you have the delta 8 there. You see that double bond move from on that right portion to the left portion now, right? And then you can have more photons come in from light, and they can actually cyclize that ring. Now that ring there is now aromatic. That's an oxidation. When you lose hydrogens, that's an oxidation, just like when you gain oxygens, that's also an oxidation, right? And then now that whole set of structures on the left with a big bracket pointing to the structures on the right, I have a delta there. Delta, that's a symbol which refers to adding heat, and you can see a CO2, carbon dioxide, popping off. So all of those acid-containing molecules can get decarboxylated. And so, yes, you can have CBNA. That's the acid form of CBN. CBN being cannabinol, which is on the right, on the bottom right there, right? The CBN's becoming of interest lately. So those are all the cannabinoids. People talk about THCV, and they talk about CBGV, and they talk about all these other things. Those are all cannabinoids that naturally the plant doesn't really make a lot of. It's just not genetically controlled. So all the plants, all the cannabis plants in the world, they either make CBD, THC, or both. And then you have some kind of anomalies that'll make more of one, and obviously selective breeding can make it so you have plants that produce more and more of just the minor cannabinoids. So, but those are the big ones. And then when it comes to the terpenes, everyone talks about, there's all these terpenes in cannabis, and there's hundreds, maybe more, around 68. There was one study that said there's around 68. But there's 4 or 5, I've highlighted 4 over here, that are the major terpenes. And those are going to be the dominant ones that are going to be in the essential oils, right? So, because we're talking about how cannabis aerosolizes, I'm going to go over some sort of basic aerosol science, physics that deals with a really interesting phenomenon called gas particle partitioning. Now, it sounds like a mouthful, but it's pretty intuitive when you think about it. An aerosol, just to define, is composed of the particulate phase and the gas phase. Particulate phase are very, very tiny droplets, and the gas phase is gases that surround these droplets. So when you look at a cloud in the sky, or a cloud of smoke or vapor, you see the cloud. It's visible to you because of the droplets. Now, those droplets are going to diffract light, and they're going to bounce it around, and so you can see them. They're visible. The gases are going to surround that. And components of that particulate phase are going to evaporate into the gas phase, and you have components of the gas phase that are going to condense into the particulate phase. And so there's an equilibrium between the particle phase of the aerosol and the gas phase of the aerosol. Different components, you could imagine the more volatile ones are going to be more present in the gas phase, and other components are going to be present in the particle phase of the aerosol. And so there's a set of equations here that are going to sort of govern this. A really important one is the partition coefficient that was developed by someone that, a principal investigator here at Portland State that I work with, and he's actually on my thesis committee, Dr. James F. Penkow. So this partition coefficient is really kind of a consequence of Henry's law of that deals with vapor pressures, right? And so when you have components that are volatile, they're going to be gases. They're going to want to be in the gas phase, and components that are not volatile are going to be in the particle phase. And you can see that Kp, the partition coefficient, is Cp. That's the concentration of the component in the particle phase divided by Cg, concentration of the component in the gas phase. And you can see that is inversely proportional to the vapor pressure. So when you have a high vapor pressure, then the component will want to be in the gas phase. It's pretty intuitive. And we can use those two set of formulas that are just below that to calculate just a fraction, a percentage of what percentage something is going to be in the gas phase or the particle phase. So, what's the consequence of this? Well, the aerosol from vaping cannabis is going to have a lot of components in it. I don't want to say an extremely large amount of components compared to like cannabis smoke, which is going to have many more. But the aerosol from vaping cannabis is going to have, for example, if you have a vape pen that has like 90% THC and 10% terpenes, that aerosol is going to be like 90% just those components, right? The THC, because it has a very low vapor pressure, is going to be entirely in the particle phase of the aerosol. You can see on my table here, I've arranged some components of the aerosol by vapor pressure. So at the top, you have the components with the lowest vapor pressure, and at the bottom, you have components with the highest vapor pressure. And you can see that the percentages in the gas phase increase, right? So the CBD and THC are going to be entirely in the particle phase with 0% in the gas phase. And then terpenes are going to slowly increase in vapor pressure, and there's more and more, you're going to have a higher and higher percentage of them in the gas phase, right? So linalool, which actually has an OH on it, maybe I can go back to this slide. Linalool has an OH on it, so it's going to hydrogen bond. It's going to be a molecule that is going to have a lower vapor pressure than, for example, myrcene, right? So linalool is going to have a higher percentage of it is going to be in the particle phase. It's only 5.3% in the gas phase, whereas pinene, for example, is 57% of it is going to be in the gas phase. So you're going to smell those things, right? And then these degradation products, toluene, methyl ketone, benzene, methacrylate, are going to be almost entirely in the gas phase because they're pretty volatile molecules. And for anyone that wants to go back and look at these slides, I just the water and the acetone lines that I gave in there just reference vapor pressures. They're pretty commonly available things, right? So you can look at that for comparison. Now, there's some pretty interesting implications in health. There's pretty interesting implications of this because, I'll start with the particle phase of an aerosol. The particle phase of the aerosols are going to deposit in the lungs. So when you inhale a THC-containing aerosol, THC is completely in the particles that little droplets, they're going to get into the lungs, and that's how that stuff gets in your bloodstream, right? But the gases are going to deposit in the respiratory tract, and it's going to depend on how soluble in water they are, right? So, because the lining of your respiratory tract is aqueous, the bronchial epithelium there, right? So, for example, methylvinyl ketone has a lower Henry's constant. That's something that's going to relate to water solubility. It's a lower Henry's constant than, for example, toluene. So that stuff is going to deposit in the upper respiratory tract, and so that's going to irritate the throat and the upper respiratory tract. The methacrylate also is something we identified, but the benzene and toluene are going to be lower, right? So these are some of the things that are implicated in like the harshness of a vaporizer and smoking as well, right? So I want to now talk about some of the experiments that I started doing when I joined Portland State here. So this is something from a paper that we published in 2017. You can see it's pretty interesting experimental setup here. There's a little dab rig, which we call it a water pipe with a ceramic nail on it, and we have a cold trap and an impinger with NMR solvent. And so when we do a dab, and at this point we're just working with terpenes because we didn't have a DEA license yet, and so we just measured some degradation products here. I'm not going to talk too much about this stuff because we built on this a lot. So I'll talk about more about that stuff, right? But I do want to point out that we were able to really accurately measure the temperature of the nail. So we had some interesting temperature-dependent things here. We heated the nail up to really hot and then let it cool down and then did the dab at the temperature that we wanted it to. So we weren't doing dabs at 590 Celsius. That was just sort of a reference temperature there. And as you might have seen this paper we posted in this journal, ACS Omega, part of the American Chemical Society, and it's the most viewed paper in that journal. Is almost 63,000 views. So it made a big splash in the dabbing cannabis world, right? And most notably, we found some really interesting chemistry with the terpenes. And what we found is that, sort of unsurprisingly, terpenes all react in a similar manner to each other. And all the terpenes, well, those three terpenes that we studied, the linalool, limonene, and myrcene, and we studied caryophyllene as well, they all react to form sort of large amounts of isoprene. If you're familiar with some OChem, terpenes are made up of isoprene units. That's kind of what defines them. So when these things react, they're going to make a lot of isoprene. And isoprene itself, we theorize, would go on to make these other degradation products. I'll explain that more in a second. And THC, you see that red highlighted part there, that should look pretty similar to limonene, right? It's because THC is a terpenophenolic molecule. It has a terpene part to it and a phenolic part. The phenolic part is that benzene ring with the OH sticking off of it. So we theorize that THC might make very similar degradation products to terpenes. And so we had to wait to get our DEA license. We got it, we started studying THC. But I want to talk more just briefly about that isoprene oxidation, because we looked into the literature and said, well, how is isoprene going to react? And what we found is that in the field of atmospheric chemistry, this is actually a pretty well-studied set of reactions because isoprene is going to be released in forests, right? And so that isoprene gets up into the atmosphere and it's going to react with oxygen in the air. And it's going to make this slew of degradation products, which is very similar to the degradation products that we observed from dabbing terpenes and THC. And this is an interesting implication here. Another side note is that there's this study that was published. I'd really recommend checking this paper out. It's open access. If anyone wants to check it out. Done by one of the people on that paper is a collaborator of ours. They looked at the air quality in cannabis grow rooms outside and in. And so there's a lot of really interesting details there. One of the major takeaways is that the better the ventilation was, the better the air quality. So that's something you want to keep in mind, because that stuff, all those volatile organics, just like the terpenes, are going to actually make ozone, and that might not be good either for anyone inhaling it, but it also could degrade, could further oxidize some components of whatever you have in there, right? So, so when we got THC in the lab, we wanted to study this as the best method that we could. And so one of our collaborators here had this really interesting experimental setup called adsorption, thermal desorption, gas chromatography, mass spectrometry. And so what this is GCMS, if you're familiar with that already. This is a sampling method for that allows you to just basically collect gases onto this little tube that you can see there, the ATD cartridge, a little cartridge full of absorbent material. Gases get stuck to that, and then a machine is going to desorb them, and then they're going to go right into the GCMS, essentially. And so that ATD cartridge can only handle gases, and so you need to filter the aerosol of all the particulates. And so that on this picture here where it says CFP holder, that's a specialized filter called a Cambridge filter pad. It's in that holder. Is going to filter out the particulates of the aerosol, right? And so what's interesting then is that when we see a chromatogram from dabbing THC, we don't see any THC in here because this is just the gases of the aerosol, no particulates. So this is just a way of looking at the degradation products. And so at the lower retention times, we see some C4 to C5 components. There's acetone, there's alene, and a lot of isoprene, unsurprisingly, because of how THC is a terpene. And then we start to see C6 and C7 as we go up in retention times. We see toluene, we see benzene, methacrylin, methylvinyl ketone, crotonaldehyde, butanal. Those are nasty things you don't want to be inhaling. And then there's all these alkenes in there that toxicologically are not too relevant, but they could be sensitizers. And then at higher retention times, you start to see less and less, but you do see some xylenes and other C10 components. Now, that's just from dabbing THC, but remember now, terpenes are going to be heavily present in the gas phase of the aerosol. And so those large peaks in the green portion there at around 20, those are terpenes, right? So when you dab THC and terpenes, those terpenes are going to get in the gas phase, and you're going to see them in this chromatogram. And we were able to quantify some of these degradation products that were present in the aerosol. We have methacrylin, benzene, toluene. These are all things we made a calibration curve for. And you see here at the top, you see the THC dab and mock distillate dab. Mock distillate was, we purchased analytical grade THC, and we added terpenes to it to make what we call mock distillate. And this was 90% THC and 10% terpenes. And you can see there's a really significant increase from just dabbing THC to dabbing THC and terpenes, a large increase in the degradation products. So terpenes are going to be much more thermally labile, which means much more prone to degradation than just THCs. They're smaller molecules. They absorb a little bit of energy, and they're just going to start breaking apart. With THC's larger molecule, it's going to take more energy to break it apart. But THC still makes, and then we also vape them. We also vape this mixture, this 90% THC, 10% terpenes mixture, put it in a standard, like a C-cell cartridge, and we vaped it at 3 different voltages. And you can see there that as you increase the voltage, that's an increase in energy applied to the distillate, and we observed larger amounts of degradation products. So these are pretty nasty things here. So I would encourage everyone to check out that paper and to sort of consider, what it is they put in a vape pen. We're going to have some more results about some of these components, for example, MCT oil and other things that people are putting in there. We're going to study that. And we encourage everyone to really think about, what it is they're vaping. Definitely, the more terpenes you put in there, likely is more degradation products that are going to be in there. Because a lot of people actually say that, if you decrease the viscosity of your oil, then that's going to lead to less degradation. Well, the terpenes degrade much more easily. So it's probably not a good idea, especially, and for really anyone that's vaping is going to be exposed to this. So people that are more sensitive to these components, they might want to watch out. Obviously, we started talking about this stuff before the whole ElSohly outbreak. This is the work that we did around a year ago, and we published it right in the middle of the ElSohly outbreak. And so everything changed after that. But it's still holds. It's getting much more interesting now with all these developments about the other additives people are using. So that is something we're going to study as time goes on. So I want to thank the National Institute of Health and the FDA for funding, and Portland State University also for funding. The Drug Enforcement Agency helped us with the security requirements necessary for us to legally study Schedule I cannabis in a lab, because our lab is fairly funded, so we obey federal laws. And the Pankal lab, Dr. Wentai Luo and Kevin McWhorter for helping us out with the ATD GCMS collection methods, and my research assistant, Alicia Ortiz, and obviously my principal investigator, my PI, Dr. Robert Strongin. All right, thank you.